Penicillium introduction and occurrence. Penicillium is a saprophytic fungus, commonly known as blue or green mold. According to Raper and Tomb 1949, the genus includes 136 species, distributed throughout the world. They are present in soil, in air, on decaying fruits, vegetables, meat, etc. The wonder drug, penicillin, was first discovered by Sir Alexander Fleming at St Mary's Hospital, London, in 1929. During his work with a bacterium Staphylococcus aureus responsible for boil, carbuncle, sepsis in wounds and burns etc., get contaminated with mold spore Penicillium notatum, which after proper growth causes death of S. aureus showing lytic zone around itself. He isolated and called this antimicrobial compound as penicillin. Later Raper and Alexander 1945 selected a strain of P. chrysogenum, more efficient than P. notatum, in the production of penicillin. The importance of penicillium is mentioned in the table below. Vegetative structure of penicillium. The vegetative body is mycelial. The mycelium is profusely branched with septate hyphae, composed of thin-walled cells containing one to many nuclei. Each septum has a central pore, through which cytoplasmic continuity is maintained. Some mycelia grow deeper into the substratum to absorb food material and others remain on the substrate and grow a mycelial felt. The reserve food is present in the form of oil globules. Reproduction in Penicillium. Penicillium reproduces by vegetative, asexual and sexual means. Vegetative reproduction. It takes place by accidental breaking of vegetative mycelium into two or more fragments. Each fragment then grows individually like the mother mycelium. Asexual reproduction. Asexual reproduction takes place by unicellular, uninucleate, non-motile spores, the conidia, formed on conidiophore. The conidiophore develops as an erect branch from any cell of the vegetative mycelium. The conidiophore may be unbranched or becomes variously branched. Rarely many conidiophores become aggregated to form a club-shaped fructification called choremium, which develops conidia known as choremiospores. During the development of conidium, the tip of the sterigma swells up and its nucleus divides mitotically into two nuclei, of which one migrates into the swollen tip and by partition wall the swollen region cuts off from the mother and forms the uninucleate conidium. The tip of the sterigma swells up again and following the same procedure second conidium is formed, which pushes the first one towards the outer side. This process repeats several times and thus a chain of conidia is formed. The conidia are oval, elliptical or globose in structure having smooth or rough outer surface and of various colorations like green, yellow, blue etc. After maturation, the conidia get detached from the mother and are dispersed by wind. On suitable substratum, they germinate by developing germ tube. The nucleus undergoes repeated mitotic division and all nuclei enter into the germ tube. The septa formation continues with the elongation of germ tube and finally a new septate branched mycelium develops. Sexual reproduction. Sexual reproduction has been studied only in few species. It shows great variation from isogamy, oogamy to somatogamy. Most of the species are homothoric, and few are heterothoric. Ascocarps are rarely formed. Based on the ascocarps, different genera can be assigned as Uropenicillium, Talaromyces and Carpentals. Sex organs. The female and male sex organs are ascogonium and antheridium respectively. Ascogonium. The ascogonium develops from any cell of the vegetative filament as an erect uninucleate and unicellular body. The nucleus then undergoes repeated mitotic divisions and produces 32 or 64 nuclei. Antheridium. The antheridium develops simultaneously with the ascogonium from any neighboring hypha. It is also a uninucleate and unicellular branch which coils around the ascogonium. The apical region of antheridile branch cuts off by septum and forms a short, somewhat inflated unicellular and uninucleate antheridium. With the development of ascogonium and antheridium, many sterile hyphae gradually entangle with them and finally after the formation of ascospores, the total structure becomes a round fruit body i.e., cleistothecium. The asci arrange irregularly inside the cleistothecium. The ascospores may be globose, elliptical or lenticular in shape with smooth, echinucleate, pitted or branched outer wall. The ascospores are released by the dissolution of ascus and cleistothecium wall. The ascospore germinates on a suitable substratum by developing germ tube and ultimately into a mycelium like the mother.